our session is focused on the structural biology and the drug design on ion channels. And I'm very glad to host today's workshop. And we have two renowned speakers. One is Professor Ning Yan from Princeton University, and another is David Julius from UCSF. So the first one, I will introduce briefly of Dr. Ning Yan. So probably many of you know Ning, she actually got her bachelor degree from Tsinghua in the Department of Biological Sciences and Biotechnology. And after that, she went to US, joined in Princeton University for a PhD program. And her PhD supervisor was Professor Yigong Shi, and she got her PhD in 2005, also with to Tsinghua, joined the faculty of the School of Medicine as a professor, and her she went back to Princeton as a professor of molecular biology in 2017. She is the founding Shirley Tilghman Professor of Princeton, and she was a HHMI International Early Career Scientist from 2012 to 2017, and then she also got the 2015 Protein Society Young Investigator Award and 2015 Beverly and Raymond Sackler International Prize in Biophysics, and also the Alexander Cruikshank Award at the GRC on membrane transport proteins in 2016. And she was also a recipient of the 2018 FAO BMB Award for Research Excellence and 2019 Weizmann Women and Science Award. She was elected as a foreign associate of the U.S. National Academy of Sciences in 2019. And today she's going to talk about how is electric signal generated, about the structure and the mechanism of the sodium channels. Welcome, Ning.
Sorry, I was on mute. Let me share my screen again.
Yeah.
Thanks, Hongwei, for the introduction. So when I was preparing for this seminar, I realized that this is a very special seminar because it's the first time that I ever present to my Tsinghua colleagues and students as a professor from Princeton. And I also am very grateful that Hongwei and his colleagues arranged my talk to April. April is really the celebration month for Tsinghua, for the anniversary, right? And also for our department, Department of Biological Sciences and Biotechnology. Now it is the School of Life Sciences. So and also for the title, just to echo what Hongwei said, there and back again. So I have been following a a ping pong trajectory. Tsinghua, Princeton, Tsinghua, Princeton. Although I don't know where my next stop would be. For sure, there will be another stop. Maybe, I don't know in how many years. All right. So as Hongwei just mentioned, since 2007, my lab has been focusing on the structural mechanistic investigation of membrane transport proteins. So because I have prepared too many slides, I realized, so I will skip this introduction, but just simply put, membrane transport is the essential physiological process. And by selecting, the physiologically and pathophysiologically important transport proteins, including both channels and transporters, we try to reveal their structure, their architecture, to understand their mechanism. And by working with different families of channels and transporters, we wish to elucidate the physical and the chemical of membrane transport, this essential process. So prior to 2014, my lab has been mainly using X-ray crystallography to study the structures of the of the transporters exemplified by glucose transporters. So as shown here, we saw like a human GLUT1, GLUT3 in 3 different functional states, which allow us to reveal the working cycle, like it's known as the alternating access cycle of cellular uptake of glucose. But there is another very intriguing event that my lab has been working on in the past decade, well, since 2007 actually, almost 14 years. So we know that electrical signaling is essential, really essential for the long distance and rapid response. So I always give the example that if you touch a cup of hot water or a flame, you can immediately withdraw your finger, right? Because this occurs extremely fast, mediated by electrical signals. If this event is this process is mediated through chemical diffusion, it may take days for you to sense that, and take another equal, like 1 or 2 days to control your finger to withdraw. And by that time, probably the finger would become like charcoal. So this just to illustrate how important electrical signaling is. Like you can hear me, I can present. For each heartbeat, for the motion, all these physiological processes rely on the very sophisticated process of initiation, propagation of electric signals. So shown here is a highly simplified diagram of action potential. I will not go into details, but just like for those audience who are not familiar with neuroscience, so in our muscles or neurons, the membrane has an electric field across it is known as the membrane potential. So just keep in mind, it is negative inside at the resting state. This is due to the asymmetric distribution of ions, mostly of sodium and potassium ions. So, but this electric field is not static. It fluctuates in response to different stimuli. So when it goes up, we call it depolarization, all right? So when it depolarizes, when this membrane potential depolarizes to certain threshold, the sodium channels, the voltage-gated sodium channels, which I will refer to as NAV channels, activate, which it leads to a rapid influx of sodium ion corresponding to this rapid rising phase of action potential. And immediately upon its activation, it inactivates through a process known as inactivation. Meanwhile, voltage-gated potassium channels activate to allow for the efflux of potassium ions, and eventually that leads to the reset of the membrane potential back to resting state. But this is a highly simplified diagram. I mean, it's subject to very complicated regulation. There are some other variations depending on the tissues. So from my description, you can tell how important the sodium and potassium channels are. So I have been focusing on sodium channels, and because of their physiological significance, the 9 subtypes of sodium channels, NAV 1.1 to NAV 1.9, are mutations or misregulations of these channels are associated with a broad spectrum of these disorders, depending on their specific tissue distribution. I will not go into details for each one. Among this, NAV 1.7, NAV 1.8, and NAV 1.9 are known to be associated with pain disorders. So here's the story. Actually, before the pandemic, David probably know that the opioid crisis has always made the headline. In fact, opioid overdose crisis has been claimed as a national crisis in the United States, and has claimed about like 130 people every day. And this also represented a huge economic burden, as shown here, like up to 78.5 billion a year. I mean, of course, it's not direct cost, but it's associated with the opioid crisis. But what's it for? It is actually for the relief of chronic pain. Therefore, there is an urgent need for alternative painkillers. And in 2006, there was a report in Nature. So a boy from Pakistan was identified who had no sensation of pain at all. So not only him, but I think several of his cousins also had this insensitivity to pain. And genomic analysis pinpointed the mutation to one gene, SCN9A. And if you remember, this actually encodes the channel NAV 1.7. And that was in 2006. In fact, 2 years before this report, several groups had independently reported the mutations in the same gene, SCN9A, that or associated with extreme pain syndrome. So now you can see loss of function of this mutation usually is nonsense mutations or associated with pain insensitivity, and other mutations that lead to probably higher expression or abnormal activation of the same gene can cause extreme pain disorder. So this evidence, this genetic evidence, has made NAV 1.7 a target for the next generation analgesics or painkillers. And because the 9 subtypes of sodium channels are so closely related, when you try to develop inhibitors targeting NAV 1.7, you want to make sure its function is specific. It will not suppress your heartbeat. Right. That's why we set out to solve the structures of different subtypes of sodium channels in the hope to identify subtype specific druggable sites. And so sodium channels actually belong to a superfamily known as the voltage-gated ion channels that also consists of calcium channels, potassium channels, and even TRP channels, although not all TRP channels respond to membrane potential. And so as a structural biologist, I have 2 very fundamental questions to address. So first, what determines the ion specificity, the ion selectivity, right? So why sodium channels only permeate sodium, but not the other monovalent potassium or calcium, which has a very similar size with sodium. And a more intriguing question is, how do these channels detect the fluctuation of membrane potential and respond accordingly? So prior to structure determination, sequence analysis of the voltage-gated ion channels have already shown that they contain 4 repeats or 4 subunits. So that made them either homotetramer, homodimer, or just one polypeptide chain that contains 4 homologous repeats. And within each subunit, there are 6 transmembrane helices known as S1 to S6. And among these 6 segments, the 4 sets of S5 and S6 enclose a central pore that is responsible for the selective ion conduction gating. And the first 4 segments, S1 to S4, each constitute a so-called voltage sensing domain or voltage sensor. And among this, S4 is the most unique one in that it carries repetitively occurring positive charge residues like lysine and arginine. So these basic residues are known to be the gating charge residues. So our knowledge on the structure and mechanism of voltage-gated ion channels actually first came from the study of potassium channels by the pioneering work of Ron McKinnon, the Nobel laureate in 2003. So shown on the left is is Nobel winning structure actually, but it is only half of the pore domain. And this is the side view. And on the right is the top view or extracellular view of the voltage-gated potassium channel. As you can see, 4 voltage sensors surround a central pore domain. So now, like, just to summarize what I've told you, like, for a voltage-gated ion channel, we know it requires at least 3 elements. You need a selective filter to discriminate different ions. You need the intracellular gate or activation gate to control the opening or closure of the channel. And you need a voltage sensor to detect the membrane potential change. So these are the 3 elements, but in different types of channels, they are different. The details are different. And in contrast to the relatively rapid progress in the structural study of the homotetrameric potassium channels, that of the sodium-calcium channels has been slow. Why? Because they are made of a single chain, and they are subject to heavy glycosylation or phosphorylation, which made them heterogeneous. So it is extremely difficult to obtain the recombinant protein for crystallization. Therefore, for a long time before 2017, we had no idea about the structure of eukaryotic single chain sodium or calcium channel. So the shown here was one published in 2000, but in retrospect, this low resolution negative stain EM reconstruction really didn't reveal anything, and it was even misleading. But prior to the study of eukaryotic channels, there were several structures of bacteria sodium channels, but they are made of homotetramers. My lab also contributed. That's our first publication on the sodium channel. But I will not go into details. Just to cut the long story short, the knowledge derived from the bacteria sodium channels, which are homotetramers, or derived from potassium channels, which are also homotetramers, cannot explain, several fundamental questions that are unique to eukaryotic sodium and potassium, and calcium channels. So just to list them, they are made of single chain. So the very fundamental question, how are the 4 repeats organized, remain unclear actually, because theoretically, if they all observe a domain swapped arrangement of the pore domain and voltage sensing domain, there can be at least 6 combinations. And also, among all the voltage-gated ion channels, the sodium channel is most unique in that it contains 4 distinct residues that control ion specificity, DEKA. How these 4 residues collectively select sodium over other ions also remained unclear. And, because this channel is made of single chain, it was already shown through biophysical approaches that they do not activate simultaneously. In fact, the first 3 repeats activate faster than the last one. And activation of the last repeat 4, actually leads to another important process known as the rapid or fast inactivation. So as shown here, this is a typical recording that, once the channel activates, it can inactivate, within just 1 millisecond. This is important to ensure, action potential. It is like a wave. This is important to ensure the repetitive occurring of the firing. So how is the channel achieve fast inactivation? Ever since 1970s, it was already shown by Armstrong and Bazzania. So it's Clay Armstrong and Pancho Bazzania. My pronunciation may not be quite right. That the intracellular segment controls the inactivation, and then later on, Bill Catterall showed that a small cluster containing 3 hydrophobic residues, IFM, as leucine, phenylalanine, and methionine, shown here actually as this ball, executes Fc activation. It is on the linker between repeat 3 and repeat 4. So this model was referred to as ball and chain model. It was quite neat. Unfortunately, this model actually contradicts with a number of experimental evidence. So exactly how sodium channels achieve Fc activation also remained to be elucidated. And this slide may be interesting to David, right? Because you study a lot of toxins, and sodium channels are actually the primary targets for many natural toxins from the animal venom. And in fact, before the structure was available, 7 sites for different types of toxins, either small molecules or peptide toxins, have been identified and mapped to the primary sequence. Unfortunately, these sites cannot be recapitulated on the model of potassium and bacteria sodium channels. Therefore, a structure of eukaryotic sodium channel is really required to understand the action of these toxins, and also local anesthetics, some FDA-approved drugs. So, okay, this is the end of my introduction, 20 minutes. For a long time, we have been struggling with the structure, expression, and production. So I will just, cut a very long story short. So since 2012, I'm really grateful that I had the privilege to work with the young scholars listed here, not all of them, but the major contributors to the study in Tsinghua. We were able to solve the structures of a number of sodium channels. So, but prior to that, because of the fantastic cryo-EM facility that was initiated in 2009, way before the revolution of cryo-EM, we were able to solve the structures of a few much larger channels like the renin receptors and the multi-subunit calcium channels. So during this process, we also trained ourselves to get familiar with cryo-EM. And then, we began to attack our top target, the sodium channels. So they are much smaller. They have a pseudo 4-fold symmetry. Technically, they are more challenging than the renin receptor, which are gigantic homotetramers, or the CAV1.1. So these are a few structures that we've determined in the past since 2017, but the struggle started in 2012, in terms of eukaryotic sodium channels. So just, I know in the audience, quite some of you are students. So I like to share some technical aspects, although it will be pretty fast. So the first, as I said, bottleneck for structure determination was to obtain, the proteins that are suitable for cryo-EM imaging. And for a long time, we've been struggling with human proteins. And then a very talented graduate student, Huaizong, who is now a principal investigator in Westlake University, came up with an idea. It was it's so difficult to get these proteins. Maybe it's because of the intracellular domains. Why don't I just look for some short isoform among all the eukaryotic sodium channels? And, with this, he actually just searched all the data bank for all the deposited sequences of sodium channels and identified one from American cockroaches. So we named this one NAVTAS. S stands for short. And indeed, now we can see a decent peak on SuperDAX. And also the peak looks decent, right? But if you really calculate the yield, it was very low at, in our opinion at that time, only 50 micrograms out of 8 liters. But now, after we've been working with human proteins, this was really a high yield channel. But this was only the first step. After we got the protein, fortunately, this protein can easily get into the vitreous eyes and, distributed with different orientations. So we did not have to struggle with cross-sample preparation. But then because of, its small size and because of the pseudo 4-fold symmetry, we've encountered a second barrier, like the data processing cryo-EM. It really took Qiang Zhou, who was trained by Professor Senfang Sui and was a research track associate professor in my lab at that time. And it took him several months just to figure out a way, a protocol, a new procedure to process the data. So in retrospect, his, struggle really gave us a fantastic workflow. So later on, whenever we started, data processing with sodium channels, we followed the same protocol and it worked like a charm. To cut the long story short, we were able to solve the structure of, the first structure of eukaryotic sodium channel. And as shown here, it contains like 4 repeats. And, the major part is transmembrane region. And on top, it is extracellular segments. You can see, the several glycosyl moieties were revealed. And on the intracellular side, there are also several domains that I will come back later. And then, but unfortunately, for this channel, which was from like American cockroach, it cannot be recorded. We could not obtain electrophysiological data. So this made it, hard to establish the structure and function relationship. Therefore, we switched to human protein again. So this time, we thought after the structure determination, the first one should be easy. It turns out it's not. So another graduate student and then a postdoc, Xiaojing Pan in my lab, really spent several years. She started this like in 2012. It took her, 5 years just to struggle for protein expression. She tried different systems, different constructs, different detergents, and eventually, it is actually the so-called BAC-MAM system that you infect mammalian cells using baculovirus that gave us, a decent yield, like 40 to 60 micrograms per 12 liters. Also, this now, in retrospect, was still a high yield protein. All right, but after we got the protein, so this is the skeletal muscle isoform is known as NAV 1.4, but it is so difficult to get this protein into the vitreous ice. As you can see, they tend to either aggregate in thick ice or like you see no particle in thin ice. Xiaojing really struggled for a long, long time. And one day I was just like, discussing with her, I was like, hey, come on, we were trained as crystallographer, right? We're not strangers to screening methods. Why don't you get a crystallization kit, which was shelved for a long time, and see, what conditions you can add as additive? So, Xiaojing really took my advice seriously. So she screened, she made a matrix to screen like salt concentration, different salt and, different pH. She started with pH. And to my surprise, actually, this was an important factor. So this shown here are SuperDAX, like chromatograms. And you can see, at different pH, the width of the half peak is different. Although, for most people, this may be nothing, but for well-trained biochemists, this means a lot. That really reflects the sample quality, whether they are homogeneous enough. And also, so Xiaojing just selected pH 6.0 to be the optimal one. And even at this specific pH, she tried different buffers, but different chemicals as buffering regions. And turns out imidazole improved the particle distribution. And then after we identified this condition, we searched literature. Turns out there was already report that imidazole could also help improve the cross sample property in another case as shown here. But then we still had some problem with the probably air-water interface. So Xiaojing and other junior students also screened for different, like, even cross-linkers or like different types of grays and also some surfactants. Eventually, addition of extremely low concentration of SDS helped to further improve the cross sample quality. So eventually, Xiaojing obtained a very decent electron micrograph, and solved the structure of the first structure of human sodium channel, which has been very well characterized. Therefore, we now have a model for structure and function relationship study. So, after this, kind of endeavor, I coined a term, proteinality. So that means every channel has its own specific, unique proteinality, just as like every person has your own personality. All right, but so after that, we solved several other human channels, sodium channels. To our probably disappointment, so far among all these channels, we only captured two major conformations. One represented by the first nav-pass structure, and all others exhibit very similar conformations. The following discussion will be based on this two conformations. So what have we learned from these structures? Remember, the first question, about selective filter. Sodium channels contain 4 distinct amino acids, D, E, K, A, to determine the sodium selectivity. So prior to structure determination, we were thinking probably this lysine would alternatively form, interact with D or E, to achieve some sort of alternating access in the select filter vestibule. However, to our surprise, after we solve all the structures, this lysine is extremely stable. That means it does not move at all. In fact, when we collaborate with Haipeng, a professor, now a professor in Tsinghua, and his student, Juanrong Zhang, we identified a potential permeation path for sodium through the select filter. So we also identified a relatively stable sodium coordinating site, constituted by DE and another invariant E in the second repeat, as shown here. So this molecular dynamic simulation performed by Juanrong showed, this permeation path. And just a few months after this discovery, we actually solved the high-resolution structure of sodium channel at 2.6 angstrom. And this time, we could see a density that very much likely belongs to a sodium, as you can see. Indeed, it is it is just consistent with the computation. So this DEE site provides a relatively stable coordination of sodium, and during the permeation, the lysine and alanine in repeats 3 and 4 just stay stable. It seems like their role is just to repel this cation to the other side. So that's our current understanding of the selective filter. And in terms of intracellular gating, because we only obtained 2 conformations, unfortunately, the diameter of their intracellular gate varies a lot. So at the beginning, we thought we've already captured a kind of activated channel with a radius of about 2.7 angstrom. But then also through MD simulation, we realized that this is not wide enough for a hydrated sodium ion. So far we haven't captured an open state. Nevertheless, the conformational change between these channels gave us a sense of how the pore domain may undergo conformational changes to open this gate. As you can see, this helical bundle undergo an iris-like rotation. Each helix S6 also undergoes axial rotation. And during this process, shown here is a surface view of the pore domain, side view. You can see actually on the side, the wall also seems like it's gated, right? So if it opens, it's called a fenestration. And, in that pass, there's no fenestration at all, whereas in other channels, we can see these big fenestrations. And also this site, this particular one, enclosed by the repeats 3 and 4, was already known to be, the binding site for local aesthetics. So therefore, this shows that, the pore domain also undergo conformational changes. This may represent potential druggable site. for subtype-specific inhibitors. All right, so the next part is my most favorite part about the FASN activation. So just let me remind you again. So this channel undergoes, quick inactivation upon its activation. And so the previous model was like a ball and a chain model. So this model was originally proposed for potassium channel and also borrowed by sodium channel. And until last year, there was no structure of a ball and chain inactivation of potassium channel either. And last year, Karina Nimijan's group from Weill Cornell actually solved the structure. And you can see this is the so-called ball. It inserts into the pore domain from the intracellular gate, right? But after we solve our structures of sodium previously, the calcium channel, to our surprise, this 3,4 linker, which carries the FASN inactivation motif, actually in both structures, it is sequestered by a globular C-terminal domain. And the IFM motif is placed really far away from the intracellular gate. So how to reconcile this? So we were like, the previous model may be wrong. Fortunately, we did put that down in our paper. So after we solved the structure of like NAV 1.4 from first electric eel and then human, we observed a pronounced conformational change of this 3,4 linker. So this can be better illustrated in the morph. As you can see, this is the original position in NAVPAS, and the other structures, this 3,4 linker undergoes marked translocation to place the IFM motif into the pore domain. But it is not like the potassium channel where the intracellular gate is blocked. Rather, this F motif is inserted into a cavity that's outside of this intracellular gate. It's outside of the tetrahelical bundle that encloses the gate. So this reminds me of the door wedge, right? If you think about it, if you want to close a door, there can be two ways. One is you directly stand there to block it, and another is you push the door to close. And by squeezing the door to close, we have this, this channel has the advantage of avoiding leakage, right? Because, if you just block this intracellular gate, when you remove the blocker, there can be potential ion leak. So that's why we propose this allosteric blocking or door wedge model to account for FASN activation. And, this model that was first observed in the structure of electric eel sodium channel, we proposed in 2017, was recapitulated in all human channels. Also, like last year by Bill Cattrall's group, and also it is consistent with tremendous amount of mutagenic study. And recently, we systematically analyzed some disease mutations, and indeed, dozens of mutations that are associated with the lung QT syndrome in NAV 1.5, the cardiac sodium channel, are either directly or indirectly involved in the segments for FASN activation according to our door wedge model. Okay, so how long do I have? I have 8 minutes. So the next part, if you remember, I told you that sodium channels are the primary targets for toxins. So for this part, I will go faster, probably. There are two prototypical small molecule inhibitors, the tetrodotoxin and saxitoxin. So they are actually the two molecules to characterize sodium channels. So just to cut the long story short, we were really extremely happy that DM is so powerful that it allowed for the resolution of small molecules TTX or STX. These structures clearly illustrated why some sodium channels can be resistant to these small toxins. Oh, I like this movie. And another type of toxin is peptide toxin. So here, it's a clip from the video like a toxin having smell that model fish. It looks like a needle, right? To stab the fish. Actually, it is like, it injects venom into the fish to paralyze it. And the main like one of the main constituents in the venom is peptide toxin known as the mucotoxin K3A, which I will refer to K3A, which is like 16 amino acid peptides that contains 3 pairs of disulfide bonds. But for a long time, how this toxin blocks sodium channel remains unclear. So, well, we solved the structure of K3A bound to NaV 1.2, a central nervous system sodium channel, and as you can see here, so this toxin sits right above the selective filter. So when we compare this one to a previous structure of potassium channel in complex with another toxin, although toxins are different, they have different length or composition, but we identified one common paradigm. So both toxins contains one lysine that just projects into the mouth to the selective filter. So this clearly immediately reveals how the toxin blocks ion conductance. And another type of peptide toxin is known as the gating modifier. I will not go into details. Basically, we solve the structures of potassium channels in complex with different types of GMT, gating modifier toxins. I will not go into details, but just to tell you what we've learned. So for these toxins, for the gating modifier, so they require, part of this toxin requires an anchorage, either like a stable pore domain elements or the membrane. And then the other part interferes with the voltage sensor to control the movement of the voltage sensor. So that's what we've learned from the structures. Okay, I hope to spend the next 5 minutes. I will skip the summary part, what we've learned about selective filter or map the disease, mutations, like how, the structures in different conformations may facilitate mechanistic understanding and drug discovery. But just let me tell you, our current challenge. So as I said, for the so the process of voltage sensing to the gate opening or closure is known as electromechanical coupling. So to understand this process, we still need two structures for the minimal. One is the channel at the resting state. Another is the one in the activated states. But for resting state, we've tried. You cannot imagine how many mutations or chemical modifiers we've tried. But the structures always remain the same, like the inactivated states. So we were like, why don't we invent some new method just to apply electric field to the channel directly. So when I moved to Princeton in 2017, this has been my major research aim, to invent a new method for cryo-EM study, to create more conditions for sample preparation. So to introduce a membrane potential or transmembrane electric field, there can be two potential strategies. One is you embed this protein into a liposome, and you try to manipulate the ion distribution, just as in the cell, right? Cell use ATP pump, sodium potassium pump, to maintain the asymmetric distribution of sodium potassium ions. So that's one strategy. Another strategy is you directly apply a physical electric field, through a device to your sample during this plunge freezing. So we actually followed both strategies, and we are still in the middle of this endeavor. So for liposome embedded protein, there are several, again, technical bottlenecks. So first is how can you, reconstitute structure out of proteins embedded in liposome. So Xia Yao, who got her PhD with Professor Yu Rao in School of Pharmaceutical Sciences in Tsinghua, actually has been focusing on this. I will not go into details. Basically, she figure out she refined the protocol, and she were able to obtain beautifully distributed liposome on the cryogrid. But during this process, we realized that we need graphene grids, and then for even distribution, of the liposomes. But for graphene grades, so for a long time, we could not obtain high-quality graphene grades from commercial resource. So actually, my first postdoc actually recruited to Princeton was Dr. Yimo Han. She actually got her bachelor's degree from 数理机科班 in Tsinghua University. She is now an assistant professor in Rice University. So she got her PhD in nanomaterial science from Cornell University. And when she joined my lab, we have been focusing on, the generation of high quality and easy to use graphene grades. So we published, we deposit like the video to YouTube, and also, in the PNAS website, you can find a detailed protocol of how to make this high quality, high yield, and cheap graphene grades. So basically with this, we were able to, obtain high quality cross sample, of like membrane proteins embedded in grades. And then Xiao Fan, who actually spent 9 years, or even 9 years in Tsinghua. He was trained by Professor Hongwei Wang for a PhD study, and work out the workflow for data collection of membrane proteins embedded in liposome. So we used ACRB as a prototypical protein as a 2 molecule. So the reported structure in that paper was like 3.8 angstrom, but now it can reach like 3.0 angstrom. So with this, the advantage is we could even observe like the curvature of the membrane. So we think this method, although we haven't applied an electric field, this may help the study of membrane proteins that interacts with the membrane for like either mechanical, through mechanical force, or like its conformation is impacted by the transmembrane ion gradient. So we are now in the process of introducing an ion gradient to the system. So all right, so maybe for long run, our goal is not maybe. So our long-term goal is to reveal like the structures of high-resolution structures of sodium channels in vivo to to elucidate its distribution in the neurons, and also to understand its crosstalk with other, among themselves and with other proteins or channels. So that's my long-term goal. With that, so as you can tell, really, my heroes for these achievements are all directly or indirectly related to Tsinghua. It's really a Tsinghua team, either my students in Tsinghua or like all the alumni of Tsinghua. I think, this is probably a good time. I hope like, this presentation is some sort of celebration to Tsinghua for its 110th anniversary, and also to our department, or now School of Life Sciences, for its 95th anniversary, and also for my own class. Like I was enrolled in 1996. It's like the 25th anniversary of birthday. So shown here, when I was preparing for this slide, I found this old picture. It is of like the group from my class, and also the class just 1 year ahead of us, by 95. And interestingly, I found the 5 structural biologists. So see, the audience, whether you can recognize them. Our host, Hongwei Wang, who was my actually mentor, the mentor to our class. And also, I'm not sure whether David will mention this guy, Maofu Liao, who is now associate professor in Harvard University, and Liang Feng, who also who is a tenured associate professor in Stanford University. And probably like HIPES represent like kind of top university in the United States for undergrads. This is someone that you may not be able to recognize. And this one, Yanhui Xu, who just recently published a beautiful structure, gigantic complex regarding like initiation transcription. He's now in Fudan University. Then I realized, among this huge group, only this small area, like we were concentrated in this small area. So this kind of polarization of memory analogy. So if, in the audience, the students are postdocs, if you want to become professor in structural biology go to Donglingshan and find this spot to take a picture. Maybe in 20 years, you will give a similar talk, not similar, of course, fancier talk in structural biology. So with that, thank you so much. Thanks again for this great opportunity. Take this opportunity as part of the celebration to Tsinghua and our department. So I'm ready to take questions.
Thank you, Ning. That's a wonderful talk, and especially a lot of memories, which is very important based on the channels. Yeah. So we can take some questions. I see there's already a question from the from chat room. First one, I guess it's about when you're talking about the reaction of animal, asking about trees law.
Lightning. They
move really slow no matter what happens. So what happened to their neural system?
So we see a fan of like a Zootopia, like the sloths. Yes, but I think it's under control whether they move faster or slower, right? Although you think they move slow, but keep in mind their hearts still beating, right? In terms of electric signaling, it's still fast, regardless how slow their motion is. And the second question, is there any rationale about screening condition for cryo-EM sample prep? I think so. As I said, when we realize, the protein yield is low, we attribute it to folding. And in order to make the folding easier for our target, we set out to look for shorter subtype that may contain, less flexible segments. So this worked. I think this actually follows rationale. And for water-air interface, because this is attributed to water-air interface, and at that time, we were not quite familiar with graphene grids yet. Otherwise, we would have tried graphene grids, but we did not have high-quality graphene grids. But because it was kind of air-water interface, so we tried like the surfactant, some detergent, so that also helped. And Junjie, hey, congratulations, Junjie, for your new chapter in your career.
The major question is following two, about the evolution of the nerve channel.
So the first question is, any evolution knowledge about how the sodium channel get the 4 repeats? Is there a crosstalk similarity between sodium and other channels on the gene side, or share the same ancestor? That's a good question, because we know bacteria sodium channels are homotetramers, and potassium channels are also homotetramers. So there was quite some debates about which came earlier, sodium or potassium. But for sure at the beginning, or not for sure, because we have we know there are bacterial homotetramers, so probably sodium channels also started with the homotetramer, and then because it requires 4 distinct residues for sodium selectivity, probably at the beginning you can imagine subunits with different residue at the select filter assembled together. But then during selection, I think it's because of this asymmetry that's required for sodium selection that eventually a single chain has been evolved. So that's my naive interpretation about evolution. And about crosstalk, yes, for sure there are crosstalk, but compared to calcium channels, our understanding of sodium channel within the membrane is still limited. It was shown that, it was suggested that the sodium channel may be able to dimerize, homodimerize, but there was no solid evidence. And they certainly interact with a lot of proteins, the tethering factors, so that remains to be elucidated. The second question is why life chooses to mainly use cation, but not anion channel for cell membrane signaling. I guess it's because of the abundance of ions. Very interesting. The other day, Chen Song from Peking University discussed with me about why there was no specific lithium channel. And I was like, maybe it's because of, at the beginning, on Earth, life we don't know, but probably life came from seawater, right? If you not calculate, but at least now, from our current understanding of the ion concentration in ocean, like sodium is most abundant, followed by potassium and calcium. And of course, there are also chloride, maybe because like chloride is dominating. There's this one specific one, and also because you study RNA and think about the charge DNA and RNA carries, right? So chloride may be required for, the electric shielding. So I think these are interesting questions, but I believe in selection during natural selection during evolution. So I think mostly it's because of the ion concentration abundance, as well as the impact of the kind of the charge within the cell. Take DNA and RNA into consideration. Next question, how to improve the map quality of the gate modifying toxin bound to sodium channel to reveal more detailed binding sites? That's a very good question. So our current, as I said, like my group's understanding of this is that it requires, so far we've solved the structures of sodium channel in complex with, I would say, 3 types of toxins. Like for Pol blockers, they are very well resolved. And for the modifier, getting modifier toxins, like as long as you can identify its rigid scaffold to stabilize the toxin, it is well resolved. But for those that require membrane, because like during protein purification, the membrane is disrupted, so the toxin became flexible. So we think the way to go, one from our attempt, we tried nanodisc. But for eukaryotic sodium channels, the protein yield is a limiting factor. For the current yield, there's no way to reconstitute the sodium channels into nanodisc. So that's why, like another Tsinghua student, Shuai Gao, who got his PhD from Liu Lei, from chemistry, actually engineered a chimeric protein with a bacteria natchback. So bacteria sodium channel as the scaffold, and grafted the toxin binding site from human sodium channels to interact with the toxins. So for that chimeric protein, we were able to incorporate that into nanodisc, and indeed the resolution was improved. But I think it really depends on the state of the channel. If the toxin prefers inactive state, maybe there's a good chance to capture a high resolution toxin. But if it prefers kind of a resting state, I guess the only solution is to get the right state, and then the binding can be highly specific, and then the toxin can be better resolved. Yeah, that's something we are still working on.
Actually, I have a question regarding the last part of your talk. By reconstitute the channels into liposome, and you showed a nice 2D conservages of the particle embedding in the vesicle, and seems that the particle the orientation is inside the vesicle. I'm wondering if you see some particle also with the orientation sitting the opposite way.
In fact, at the beginning, we thought, this protein has preferred orientation. Turns out it has preferred selection of the particles. Then, in the past year, we've been collaborating with Zhang Xianzheng from IBP, using his algorithm, to reprocess this data. We actually obtained class of higher resolution for the channel with the other orientation.
And then the problem is, if you want to apply for instance, the ion gradient onto this kind of liposome, if you have a different orientation of the particle, would that be a problem for you?
That's actually, I think this is an advantage, right? Then you can capture the channels, because classification is powerful. You can discriminate these two classes. Right, some some channels are in the kind of positive potential, some inactive potential, and luckily, if we are lucky enough, we may be able to capture 2 distinct conformations. So I'm not worried about it.
Mm. Do we have more questions? Okay, if no more question, thank you again for the wonderful talk.
Thanks.
Okay, so our next speaker would be Professor David Julius from UCSF. David actually received his undergraduate degree from MIT. Around the year that I was born, and probably before Ning was born. And then
thank you for saying that, Hongwei.
He was studying at that time in the laboratory of Alexander Ridge on the mechanism of tRNA amino acetylation. And then he moved to the University of California at Berkeley for PhD studies from 1977 to 84. He was working with Jeremy Stoner and Randy Sheckman to elucidate the mechanisms of peptide hormone processing and secretion using Saccharomyces. He then went to Columbia University, joined the Richard Axel's group for postdoc study from 1984 to 1989, where his focus turned to neuropharmacology and also receptor function. And during this time, David actually developed novel expression cloning methods that enabled him to identify genes encoding members of the serotonin receptor family. And then David joined the faculty at UCSF in 1990, where he is currently the Morris Hastein Chair in Molecular Biology and Medicine, and also the chair of the Department of Physiology. David's major focus is to elucidate the molecular mechanisms of somatosensation and pain and the sensory adaptation. And his group has exploited the properties of natural products to discover a family of thermal and chemosensitive ion channels. Many of us actually now know on the TRP channel, which is very important. And also about 8 to 7 years ago, David's group collaborating with Yifan Chen at UCSF, they use cryo-EM to first time solve the structure of the TRPV1, and that's actually a landmark work of to starting the resolution revolution in cryo-EM. And this is really a major change of the structural biology. And David himself is a member of the National Academy of Sciences, the National Academy of Medicine, the American Academy of Arts and Sciences, and the Hungarian Academy of Sciences, and the Norwegian Academy of Sciences and Letters. His work has been recognized with numerous awards, including the Shaw Prize in Life Sciences and Medicine, the Paul Jason Prize for Biomedical Research, the Canada Gardner International Award, the Rosenthal Basic Medical Sciences Award, and the Breakthrough Awards in Life Sciences, and recently Kavli Prize in Neuroscience, and also the BBVA Frontiers of Knowledge Award. And today, David is going to give us a talk on natural products as probes of the pain pathway from physiology to atomic structure. Welcome, David.
Thank you, Hongwei, and many thanks to you folks for inviting me to spend some time with you today, even if it's electronically. My screen to do. And yes, I am an old guy, but we're still doing research, so it's fun to be here. All right. Does that everybody see that well?
Yes.
Okay. All right. So today, as Hongwei said, I'm going to talk to you about natural products as probes of the pain pathway, focusing, of course, on ion channels. And I think I have to go into presenter mode. Hang on a second. Sorry, guys. All right. Okay. let me begin by giving you some background. I know there are some students here, and so I wanted to give some background. And really, our work really is rooted, all the structural work we do and other work we do is really rooted in understanding mechanisms, sensory mechanisms, and in particular, for today's talk, how we experience pain. So I just wanted to start off with a sort of obvious question, why do we experience pain? And we experience pain because it's the main warning and protective system that allows us to recognize injurious or potentially injurious situations and to initiate appropriate protective reflexes. And I often say this, and I believe it, that among our senses, I think our sense of pain, somatic sensation, is in some ways the most vital for our well-being. And as Ning had pointed out, there are some people who have mutations in sodium channels or other proteins, other genes that render this system inoperative in people, that diminishes or eliminates their ability to sense pain. And those people are at great risk for death and injury. So we need this system to stay healthy. The problem is that acute pain is very beneficial to protect us. But as we all know, pain can undergo this transition where it becomes persistent or chronic, and it outlives its usefulness as a warning system and instead becomes debilitating. And one of the goals in this field is to understand how this transition occurs, so that we can reverse this process and prevent the acute and beneficial aspects of pain from becoming debilitating. So the challenge is to better define different types of pain syndromes, understand how different how are different chronic pain syndromes initiated, and what are their underlying molecular mechanisms. Ning and I we're thinking along the same lines in terms of therapeutics. We know that throughout the world, but primarily in the United States, there's been this big problem of the opioid epidemic because the treatment of pain is really limited to two types of drugs, one of which is opiates. And this has led to tremendous overuse and death and mortality and morbidity over the years, which really just highlights the need to develop different and mechanistically different types of analgesics that can be used to treat pain syndromes. So one of the goals in this field, I think, is to understand how different types of chronic pain are mechanistically and clinically distinct. We know that lower back pain is different from migraine pain, different from inflammatory pain, arthritis, and different from the sort of pain you might experience like shingles from a viral infection, so-called post-herpetic neuralgia, or itch for that matter. And understanding how these different pains are mechanistically distinct will also help us think about how we can develop pharmacologically tailored treatments to deal with different pain syndromes that at their basis must be different in terms of the molecular players and the cellular and systems networks that are involved. So that's sort of writ large in the big scope, some of the questions that people in this field wrestle with. In my lab, we've really focused mostly on the peripheral aspect of pain sensation. So pain is initiated when sensory nerve fibers in the periphery are activated by a stimulus like high temperature or low temperature, pressure, etc., or chemicals, noxious chemicals. And that information is then sent along this primary afferent to the spinal cord, where that information is sent by ascending spinothalamic tract neurons to the brain. And one, of course, can study pain at all these different levels of the neuraxis. We focused mostly on the sensory nerve fibers where acute pain signals are initiated. And the sorts of questions that we and others in this field have asked is, how many different types of sensory nerve fibers are there? What are their modalities and sensitivities? Are some sensitive primarily to heat or cold or pressure or chemical irritants? What are the target tissues for these nerve fibers? Do some innervate cutaneous sites like the skin primarily? Do others innervate regions of the meninges that may contribute to, for example, migraine pain, or to the gut or bladder to signal visceral pain? And what are the different contributions of various different sensory neuron subtypes and the molecules that they express what are their relative contributions to different types of persistent pain syndromes? So again, these are sort of the questions that really drive our motivation in this field. I want to point out before we move on that sensory nerve fibers, such as these little fibers that are denoted here, are really very beautiful and amazing machines of integrative physiology. So shown here is a sensory nerve fiber that, say, would innervate your skin and then send that information to the spinal cord. And they have in aggregate, they have a really interesting and important job, and a complex one. And that is that they have to appreciate a very wide array of different types of stimuli, including things of a physical and a chemical nature, and to transmit this information to the spinal cord. So they need detectors on their surface that can appreciate all these different sorts of stimuli. Additionally, these cells are somewhat unique, or neurons, in the sphere of the nervous system and the world of neurons, in that not only do they send information from the periphery to the central nervous system, but once activated, they also release transmitters peripherally in sort of this antidromic signaling once activated. So that is that when these nerve fibers are activated, they transmit the signal to the spinal cord and release glutamate and other transmitters centrally. But when activated at high intensity, they also release transmitters from the peripheral nerve terminal, most notably peptides like CGRP and substance P. These peptide transmitters then act on a variety of cells in the environment, blood vessels, mast cells, basophils, etc., and induce these cell types or structures to produce and secrete a whole range of transmitters, like common ones like histamine, serotonin, neurotrophins like NGF, even proteases, peptides like bradykinin, bioactive lipids like prostaglandins, etc. And all of these factors then act back on the sensory nerve terminal to enhance their sensitivity to temperature, touch, etc. And this process of activation, release of these peripheral transmitters, elaboration of what we call components of the inflammatory soup altogether, and their actions on the nerve terminal, is a process that we call neurogenic inflammation that drives the sensitization of these neurons to then heighten sensitivity of the nerve fiber after injury. And this is really an important part of peripheral mechanisms that lead to and initiate pain hypersensitivity, at least in the periphery. And what's implicit in this is that these nerve terminals must have receptors for all of these different agents. And one of the goals is to understand how they work and how all this information is integrated to change the excitability of the sensory nerve fiber after injury or inflammation to then heighten sensitivity of the pathway. And of course, the biological and physiological reason to do this is really as a protective mechanism, so that if you injure yourself, you're hypersensitive to things like temperature and touch as a way of guarding that area and ensuring that you don't injure it again. But of course, as with all things in the pain pathway, this cycle of neurogenic inflammation can exceed its usefulness and instead become overactive and enhance sensitivity to the pain pathway long after it's useful, and drive mechanisms that are involved in generating chronic pain syndromes. Now, we've asked how these neurons work, and the way that we've done this, as Hongwei alluded to, is to really exploit, in some ways, folk medicine and natural products by asking how a number of, in this case, natural plant products in our environment generate a sensation of irritancy and pain. As many of you know, the pungent ingredients in chili peppers like capsaicin, even menthol in cooling agents in mint leaves, and then these compounds, isothiocyanates, thiosulfonates from onions and mustard plants, all generate a sense of irritancy, and they do so in a way that's quite beautiful and really a beautiful example of convergent evolution. So when you come in contact with these agents, usually you eat them, but it could also be that you chop a chili pepper and stick your finger in your eye. You will experience pain, and all of these agents work the same way. Shown here for capsaicin, but it's sort of a general phenomenon. When you come in contact with these, the active ingredient, in this case capsaicin, activates sensory nerve fibers, so-called nociceptors that detect painful stimuli, the ones that we just talked about a moment ago. They depolarize a cell, lead to an action potential, which allows your brain to understand that you've just experienced something noxious. And then, as I said, they release these transmitters peripherally to set up this process of neurogenic inflammation. And what's really beautiful about this is that all of the natural products that I just mentioned work in the same way. They all target these sensory neurons. They target maybe distinct subtypes, but they target the same kind of molecules, and that is a family of ion channels that Ning has mentioned that we'll talk about in more detail, called TRP channels. So they've all converged on this one beautiful family of ion channels in this really magnificent example of convergent evolution as a way of activating nerve fibers that are involved in pain sensation. And of course, for the plant, presumably this is a mechanism to protect them from predation by squirrels or deer or other mammals that feed on them. And we as humans have learned, as usual, to appreciate things that are harmful to us or that feel like they're harmful. But this all works through activation of TRP channels. So TRP channels are largely non-selective cation channels that are permeable to sodium, potassium, and calcium ions, although some are more selective for mono or divalent cations, but many of them are non-selective cation channels. And showing the same diagram that Ning showed, this dendrogram, we can see that TRP channels represent a very large or significant component of this so-called superfamily of voltage-gated ion channels. And in fact, among ion channels, TRP channels are the second largest and most diverse family of channels, second only to voltage-gated potassium channels. So they're a big family, and really, truth be told, there are many members of this family are still mysterious in terms of what they respond to and what physiological roles they play in vivo. We've focused on three members of this family, which correspond to the receptors for the natural products that I just showed you. TRPV1 as a receptor for capsaicin, TRPM8 as the receptor for menthol, and then the molecule that I'm going to talk about mostly today, TRPA1, which we call the wasabi receptor, because it's activated by those sort of strange chemicals from mustard and onion or allium plants that belong to the isothiocyanate or thiocyanate family. So let's talk about this channel. It's really a fascinating channel, as you'll see in how it works and what it does physiologically. So, TRPA1, as I said, is activated by this range of pungent agents from all these different vegetables, essentially. Wasabi is allyl isothiocyanate. That's what's generating the pungent feeling when you eat wasabi. But phenyl isothiocyanate, for example, is found in broccoli plants. And then these alpha-beta unsaturated aldehydes are produced by garlic, for example, or so when you're chopping and you're rubbing your eyes in pain, that's because you have gotten diallyl disulfide in your eyes. And when you look at these compounds, you think, well, they don't look very similar in terms of their overall molecular structure. So how do these all activate a channel, and fit into a pocket that could be acceptable, that can accept all these different chemically diverse agonists? And the answer turns out actually be quite interesting. But the other thing I should say is that all of these compounds are many of them are volatile, and that's why when you chop an onion and experience some of these things, you feel them quite immediately in the conjunctiva in your eyes and other places. But what unites these compounds more than anything is not their chemical structure, but their chemical reactivity. They're all fairly good electrophiles, and that's true not only for activators of this channel that are found in plants and that plants use to defend themselves, but by things in our environment that we need to detect that are dangerous, that are also in some cases volatile, that warn us of impending danger. So, for example, acrolein, formaldehyde, or 2-pentanol are also alpha-beta unsaturated aldehydes that are produced, for example, during forest fires that we need to detect in order to know that we shouldn't be in that area. And then there are some endogenous compounds that we produce in our own bodies, like 4-HNE or these prostaglandin derivatives, that are also unsaturated aldehydes that are electrophiles and reactive, that are produced when there's tissue injury, inflammation, and then the production of reactive oxygen species that peroxidate membrane lipids and lead to the production of these kinds of agents. which are found actually at significantly high levels in, for example, fluid from arthritic joints or other parts of the body that experience inflammation. And so these are all warning signals. They're all chemical irritants or chemical warning signals that tell us when we've experienced injury or when we might be experiencing injury. So we really need to detect them in order to stay healthy. And the way that they all work on this channel is very unusual. And that is that the channel, shown just one subunit here very schematically, has a number of nucleophilic sites in its cytoplasmic amino terminus, including these cysteine residues that are the main locus for modification, chemical and in many cases reversible covalent modification by most electrophiles. The channel also has a whole string of ankyrin repeats, which gives it its name of TRPA1 and is part of its structure, as I'll show you. The really beautiful thing about this channel is that, as we showed years ago, if you make a mouse that lacks the channel, so a TRPA1 knockout mouse, and you subject it to any one of these kind of electrophilic irritants, for example, by injecting it into the paw of the animal, in a wild-type animal, you'll see that it exhibits what we call robust nose-offensive response. It will lick its paw and lift it as an indication of discomfort or pain. In an animal that lacks this channel, there's no response in this way to any of these agents, at least at reasonable concentrations. And what that really demonstrates is this channel is sort of it's a broad spectrum detector of many things in our environment that only have to have in common that they're electrophilic, which encompasses a lot of things that are dangerous. And this channel is sort of a integrator of all those signals and a uniform detector of a broad range of chemicals that fit this chemical profile. Now, of course, we want to know how this all works, and that's really kind of the subject of what I'll be talking about today. And a few years ago, Candace Paulson, now at Yale, and J.P. Armash, now at Penn State, worked together with myself and my good friend at UCSF, Yifan Cheng, who you've heard so much about already, and solved the structure of the human TRPA1 channel using cryo-EM. You can see this beautiful structure here. And so this is the transmembrane region, and these are all the ankyrin repeats scaffold sitting below the membrane in the cytoplasm. And this crescent are some of the many ankyrin repeats that we were not able to resolve, but which are part of the structure. And here's a schematic of what this channel looks like. Ning's already beautifully introduced it to this family of voltage-gated ion channels, and TRP channels are distant relatives of that. They have a pore domain that's formed by the S5, S6, and intervening pore loop regions. They have an S1 to S4 domain that we call the voltage sensor-like domain because it's reminiscent structurally of the voltage sensor domains of voltage-gated ion channels. It's sensitive. It does not have the charge repeats in the S4 domain that Ning talked about that are so important for conferring voltage sensitivity to classic voltage-gated ion channels. There's also this region that I'll talk about called the TRP-like domain that's somewhat unique to these channels. It's sort of a very unusual alpha helical domain that, as I'll show you, sits just at the border or just below the inner leaflet of the plasma membrane and lies parallel to it, and which functional studies over the years and structural studies now confirm is really sort of an important locus for allosteric modulation of the gate. It sits just below the region that forms the gate in the S6 domain or very close to it, and serves as sort of, as you'll see, an interaction locus that couples whatever happens in the cytoplasm or other regions or in the voltage sensor-like domain to the cytoplasmic gate of the channel, and is therefore a very important integrator for information. In this channel, as in many TRP channels, there's a slew of these ankyrin repeat domains. TRPA1 has the most of any known TRP channel, and then there's this linker region that connects all these ankyrin repeats. You know, we don't really know what many of these ankyrin repeats do in many TRP channels. They're important in some cases for subunit assembly, as in TRPV1. In the case of TRPA1, we have little understanding as to what their physiological role is, but if you try and remove any of them, the channel is dead. In this case, the channel is scaffolded together by C-terminal coil domain, which I'll show you in a minute. So here's what the structure looks like in a ribbon diagram when we put 4 of these subunits together. It's quite a beautiful structure, and here you can see the transmembrane domain. Here's the TRP domain, and you can look down the channel and see the pore. Here are the voltage sensor-like domains on the outside. And as you'll see in a moment, there's a beautiful coil-coil interaction in the center of this channel that scaffolds and holds together all the subunits. And again, here you can see the voltage sensor-like domains from the bottom. Here are the anchor and repeats that we could not resolve, but which we sort of loosely fit into this density here at the bottom. And we imagine this is what the channel looks like when all 16 or so anchor and repeats are present in the structure. Now, since we're talking about drugs, I would just mention tangentially that we did solve the structure of this channel with a sort of canonical antagonist sitting in it. And you can see that this antagonist packs in between the pore helices and the S5 domain of the transmembrane unit. And as I'll show you later, we think that what this does is to prevent a straightening of a kink in the S5 domain, which is required to transmit information to the lower gate to activate the channel in response to changes in the outer pore domain. But mostly and here's what this looks like when it's packed against the S5 region and sort of held there and wedged there between one of the pore helices and the S5 domain. But really what I want to focus on today mostly is the mechanism by which electrophiles activate this channel, because I think that's in some ways the most unique and beautiful aspect of this channel. So shown here in these little yellow balls are the cysteine residues that we and others have implicated as being important for electrophile activation just based on site-directed mutagenesis. And if you look at the location of this in this ribbon diagram, you'll see that there's this area below the trip-like domain and above the anchor and repeats, this area that we call the allosteric nexus, for lack of a better term, that contains a cysteine, one of these the most important cysteine residue of the 2 or 3 that we and others have identified, the cysteine 621, that comes at the end of this alpha helix. And Thomas Taylor Clark's group has shown, after we identified these using some beautiful chemistry, that this is a rather unusual cysteine residue because it is about 8,000 times more nucleophilic than the average cysteine residue in a protein. So because of its location in this, at the end of this alpha helix and the chemical environment, it's a very, very sensitive detector of electrophiles. So this is sort of like the canary in the coal mine for this molecule. It's the residue that allows this channel to be so sensitive to electrophiles. And the goal, of course, is to be able to detect an electrophile before the chemical concentration becomes so high that it just manifests and leads to wholesale tissue damage. So this channel needs to accomplish 2 things. It needs to be a very sensitive electrophile detector. At the same time, it needs to be a very broad scale electrophile detector to be able to detect almost any chemical entity that is electrophilic and can cross the membrane. So what we wanted to do is to understand how this works and where it's poised. And then there was some controversy about other cysteine residues that I'll talk about in a moment, and whether and to what degree they contribute to electrophile detection. Initially, we posited that C621 was was located in this polar pocket, and that somehow modification would lead to a conformational redistribution of the protein that would lead to opening of the gate. And as you'll see, at least to some degree, that's sort of true. Now, in our initial structures that Candace and John Paul solved a few years ago, we saw the channel in one state that we took to be maybe a desensitized state. The bottom gate seemed to be closed, too narrow to pass hydrated or even partially dehydrated cations. And as I'll show you, and as you may know, the upper restriction, as we call it, or the selectivity filters and trip channels, is substantially more dynamic conformationally than you would find in most voltage-gated ion channels, whereas Ning showed you there's a very stable scaffolding of carbonyl residues that holds this part of the protein very rigid and allows for high selectivity, for example, of sodium or potassium or calcium ions. In the case of trip channels, this is a much more mobile element, and there's been some questions as to whether and to what degree it actually contributes not only to ion selectivity, but to gating. What we wanted to do was to solve structures of the channel in other states, like open states, for example, and to be able to directly visualize its modification by electrophiles and understand how the electrophiles mediate conformational changes leading to gating. So here are the questions: can we stabilize and visualize a ligand at open state? The other question we had was, are there TRPA1 activators that work non-covalently, that activate the channel through mechanisms that don't involve chemical modification? And if so, do these kinds of activators elicit distinct biophysical, physiological, or behavioral effects? So Jinhua Zhao, who just moved to have his own lab in San Diego, and John King, a former student in my lab who just moved on to do a postdoc at Stanford, got together to form what we call Team 3D Wasabi 2 and take this area to the next level. And again, I should say all these studies, of course, were done with my good friend at UCSF, Yifan Cheng. And what they did was to solve this problem of trapping this channel in its open state and its liganded state in sort of a clever way. So what they did was to first react the channel with an irreversible electrophile. So iodoacetamide will make a bond to a cysteine residue, and once it forms that chemical bond, it's irreversible, as opposed to wasabi, which will be reversed by water hydrolysis. And they did this while the channel was still in the membrane, so it could go make a transition from closed to open state. And then they solubilized this and used, the standard tricks now in cryo-EM to classify these structures into what we call class 1 and class 2, which is closed and open, and then to also verify a closed state by looking for the state that bound this A96 antagonist as an index of a closed state. And the other thing they did was, since almost any electrophile will activate this channel, they took iodoacetamide and derivatized it, or you can buy this with a very large fluorescent group, BODIPI, and use that to label the channel so we'd have a better chance of seeing it attached to one or more cysteine residues in the protein. And here you can see in this allosteric nexus, indeed, at this very highly nucleophilic residue, the cysteine 621, you can see the BODIPI iodoacetamide attached beautifully to this residue. And in fact, we did see some attachment, as I'll tell you in a moment, to C665, but the bulk of the attachment and most of the species we saw had a single modified C621 right at this site. And indeed, we also were able to show that these two structures represented what appeared to be at least more closely aligned with a closed and an activated state. And you can see the big dilation of the lower gate and even an enhanced diameter of the upper gate / selectivity filter. So we take these as being activated or open and closed states respectively. And now we can look at conformational transitions in this protein that hopefully correlate. And what we saw was actually quite beautiful. And I should say that similar transitions and structures were reported about the same time from the Lee group at Duke University. So I think there's agreement as to how electrophiles work, at least to some level. And I'll talk about some of the differences in a moment. So here you can see this allosteric nexus lying close to the trip domain is right here. And what you can see in transitions between the open and closed state is this big movement of this loop, which is shown here. So you can see, I animated this a little too fast, but you can see the change in the structure of this loop, which we'll talk about here in a minute. And this is where the protein is modified by the electrophile. Let me just back up so I can go through this for a second more carefully. All right, so you see these transitions in the scope of the whole channel, and you can see these conformational movements that are associated with it. For today and the shortness of time, I just want to concentrate mostly on what's happening right here. And if you look at a blow up of this region, here what you'll see is here's the loop that's moving up and down. It's sort of like this clamshell action. And here's the C621 residue. That's the all-important residue that's modified by an electrophile. Here's another residue that was implicated by mutagenesis. That's important for at least sensitivity to some electrophiles, C665. And then there's this lysine residue called K671 that's also known to be important by structure function work. And I'll talk about that in a minute. But you can see that its side chain becomes majorly reoriented when this clamshell opens up in response to the protein being modified by an electrophile. And we'll talk in a second about what we think the structural consequences of that might be. All right. And so what we think happens is that an electrophile modifies at least one of these residues, C621. This leads to this movement that I talked about and a repositioning of this of a lysine residue right here in K671, very close now to the overlying trip domain that I told you is an important allosteric modulatory helix in the channel that lies just close to the lower or cytoplasmic gate. And in fact, we know that this transition is quite robust because in the apo structure, namely the close structure, we can't even see a density for this side chain, but once activated, it becomes a very strong density and we can see its position quite clearly. So it really is not only moved quite a bit, but it is then stabilized, and it becomes stabilized by the interaction with the C terminus of the trip helix. And this really changes the dipole moment of this helix and compared to the apo state. And the net consequence is that, and you can see here the beautiful density where you see this ionic interactions between this reoriented lysine residue and the C-terminal region of the trip helix. And what this does is to increase positive electrostatic surface potential, or at least that's how we model it, in these trip domains, leading to a repulsion of the trip domains as they come together in the central pore of the channel or near it for all the 4 subunits. And we believe that this repulsive action helps to pull apart the S6 domains that are involved in gating. So that's at least our initial model for how electrophile modification of this channel leads to changes in the gate through repositioning of this lysine residue following big movement of this clamshell, and then close apposition to this all-important trip domain, which then lies in close proximity to the lower gate and helps to activate the gate. Now, one question, so I think that this sort of solves a couple of questions. One is, how does an electrophile and chemical modification activate this channel? The other question is, how does this channel appreciate so many different types of electrophiles, both large and small? And I think the answer to that came from some structural studies that we did and mutagenesis that I don't have time to go through in detail. But what we know is that C621 is required for activation by all electrophiles, large or small. If you have a small electrophile, then you need to modify both C621 and C665 in order to stabilize this clamshell transition to bring this lysine close to the trip helix. So you need to pack 2 electrophiles in there that have modified these 2 sites. If you have a large electrophile, something like a modified lipid, like a prostaglandin or 4-HNE, or even this BODIPI iodoacetamide, which has a big molecular profile, then you only need to modify the C621 residue in order to stabilize this transition. So I think this not only clears up a question about C665 and whether it, along with C621, is required. It depends which electrophile you use to activate the channel, but it also tells us something about the architecture of this pocket and how it needs to be occupied in order to mediate this transition. And I haven't talked much about the outer domain of the channel due to time, but I just wanted to note that this antagonist, which is sort of a classic antagonist now in the field and is sort of a basis for devising potential therapeutics for this channel, particularly in the setting of inflammatory pain disorders, we think works by packing close to the S5 helix between the pore helix and the S5 transmembrane helix, and to prevent this kink from straightening out that seems to have to occur in order to coordinate with opening impinging on the S6 domain and opening of the lower gate. But we still have a lot to learn about all the conformational transitions that are driven by modification and that are driven by ion permeation to stabilize the lower gate of the channel and how all this allosteric coupling works between the lower and the upper gate. So that's sort of work for the future that will require the use of other types of drugs and things to really understand that. Now, the other thing that I wanted to mention is that, like a lot of TRP channels, TRPA1 is also a receptor-operated channel. So TRP channels were first actually discovered in Drosophila in the phototransduction pathway, and their activation of a light-activated receptor, rhodopsin, leads to activation of PLC and the cleavage of PIP2 in the membrane, which then leads to the activation of TRP channels in the fly eye. And many mammalian TRP channels have retained this sort of ancestral function, where they're activated not only by extrinsic factors like chemicals, but they're also activated downstream of metabotropic, for example, G protein coupled receptors like a bradykinin receptor that use this same kind of pathway. And for TRPA1, one of the second messengers downstream of PIP2 hydrolysis that's very important for activating the channel or regulating it is cytoplasmic calcium release from calcium stores. And in the structure, we can see that in this region of the channel, there's a very beautiful calcium binding domain that coordinates these calciums. And John showed that when you mutated any one of these residues that's involved in that we see in this structure, that the ability of the channel to serve as a receptor-operated channel is severely diminished. So this calcium site is important for the mechanism by which this channel is activated downstream of metabotropic receptors, which is probably an important pathway whereby a number of pruritic or itch-causing agents work to activate sensory neurons. This channel is also desensitized by calcium, and this same site likely plays a role in that. And interestingly, architecturally, this is turns out to be, we've learned in the last few years, a very highly conserved site for coordination of calcium TRP channels. Other TRP channels like TRPM4 and TRPM8 also have a calcium binding domain. Calcium has a lot of effects on TRP channels, including activation as well as desensitization. And this architecturally conserved site seems to be important for that kind of calcium-dependent modulation for a number of members of this family. So we don't understand how yet this is manifest in terms of conformational changes, but it at least identifies a site that's highly conserved for a very conserved physiologic function of these channels. Now, the last thing I want to just breeze through, because I thought I had more time for this, but I think is an interesting piece of biology that I'm just going to skim through quickly, and was recently published, is to ask this question. So we know that electrophiles work to activate this channel. Many of them are membrane permeant. They go across the membrane and they activate the cysteine residue in the so-called allosteric nexus, which is on the cytoplasmic part of the channel, just under the TRP domain and the membrane inner leaflet. And of course, calcium regulates the channel. But are there things that activate the channel from the outside, or are there other agonists that work that don't involve covalent modification? And so we went looking for these things. And as Ning has beautifully said, many of us in the channel field have taken advantage of other types of natural products, not just from plants, but from animals, namely toxins from animals that use these toxins and venoms to defend themselves. And in the pain world, we all know that inflicting pain is of defensive value. So much in the same way that the chili pepper plant protects itself from predation, these animals not only kill prey, but will protect themselves from predators by generating toxins that activate the pain pathway. So we've exploited this by looking through various venoms to find so-called algogenic or pain-producing agents from these venoms that might activate channels on nerve endings. And we found a bunch that actually target TRPV1, and more recently that target TRPA1. And as Ning showed, the beautiful thing about toxins is that they have millions of years of evolution, these animals, to develop molecules, small and other molecules, peptides that target the most functionally important parts of the channel. So you can learn a lot about understanding where these toxins target their channel prey. And in the case of TRPV1, spider toxins that we've used have been really valuable for locking the channel in these persistent open states, so we can really see what happens to the pore and understand a lot about how the channel changes its shape during the gating process. So John, after he helped solve the structure with Jinhua, he went on to look for toxins, and here you can see John looking at one of his prized lab mates here that he's trying to get some venom from. And in fact, he found a venom from this scorpion called a black rock scorpion that we received some venom from our collaborators in Australia. And he just used calcium imaging to screen to find this venom and then to isolate its component by just sort of standard biochemical fractionation. And he found this one toxin from the black rock scorpion that activates the channel very well. It's a toxin that we call wasabi toxin because it, of course, activates the wasabi receptor. And here's the same channel activated by wasabi itself, or allyl isothiocyanate. And it has a pretty good affinity. The thing that I'm going to skip through all the physiology and tell you because we're short on time is that the really interesting thing about this toxin that we found, which is a real surprise, is that you can apply it to either side of the membrane. So here's a channel in a patch pipette, and you can apply the toxin on the outside. You can apply it on the inside. It doesn't matter. It still activates a channel. We were expecting to find a peptide toxin that worked from the outside, as we have for TRPV1. And it was really to our surprise that we found that this toxin seems to be a membrane penetrating toxin. So that is that even when it's applied from the outside, its mechanism of action seems to be for it to cross the membrane and find a site in the cytoplasmic side of the channel to activate it. And this was really a surprise. And in fact, at first we were disappointed because we were hoping to find something that would bind to a conformational sweet spot on the outside of the channel. But when you see something really unusual and interesting, of course, you got to follow it. And it turned out that this toxin called wasabi toxin is a member of this very poorly characterized family of K-kappa toxins that are found in scorpions. And then John solved the NMR structure of it and found that it's this tightly scaffolded helical hairpin peptide, similar in some ways to the peptides that Ning talked about, in that they're scaffolded with a small number of disulfide bonds, in this case 2. But this toxin has this sort of very unique or this very rigid hairpin structure. And it's also amphipathic because it has a basically charged region patch and then an acidic region. And so we think that, and this is sort of a hallmark of so-called membrane penetrating peptides like TAT and antennapedia and other peptides that are able to associate with the negatively charged lipid head group and then translocate themselves across the membrane. And I won't go into all the details of this. And we were able to show through biochemical studies and using synthetic lipids that, in fact, the toxin biochemically can cross a membrane where it can interact with the fluorophore inside. And you can measure this by FRET. And because of the linear kinetics of this, we surmise that, like many membrane penetrating peptides, that this is not a facilitated movement. In other words, no proteins are required. And of course, this is just a bare lipid membrane, but that this is some kind of not an active, but a passive transport of the protein, of the peptide across the membrane. So it's kind of a unique toxin. And the question was, where does it act on the channel? And the way that we were able to determine this is to take advantage of something that we've learned and that other people have shown, over many years, that sensory systems evolve rapidly in evolution to suit an animal's needs. And therefore, there's often a lot more evolutionary drift in sensory receptors from among species than you would find, say, in other channels like glutamate or serotonin receptors. And so it turns out that the toxin works very well to activate the human TRPA1 channel, but not an ortholog from a snake called a Texas rat snake. And so we're just able to make chimeras and identify the confer sensitivity to the toxin. And you notice that the region that we identified contains this residue called the C621. That's the main site for electrophile action. So basically, this toxin has evolved to target the same part of the channel, this allosteric nexus that's targeted by electrophiles. Quite amazing in terms of evolution. And we don't know exactly how it binds because we haven't yet determined a co-structure of the toxin with the channel, although we're working on it. But if you map out residues that by mutagenesis we can show are important, that John showed are important for toxin binding, they sort of define this little cleft between subunits, and we're thinking that maybe the toxin pokes right in there and somehow maybe causes similar conformational changes to occur as would occur if you covalently modify C621 with an electrophile. The one thing that I want to point out from this slide that's interesting is that although the toxin activates the channel, as does wasabi, it does so in a way that's biophysically a little different because the relative permeability of the channel to mono and divalent cations is substantially different when you activate the channel with the scorpion toxin, wasabi toxin, versus wasabi itself, allyl isothiocyanate. So the channel has a lot lower permeability to, say, magnesium and calcium compared to sodium when you activate it with the toxin versus wasabi itself. And I'll show you that we can use this to understand something about the physiology. And this is the last couple of slides. And that is, so what happens when you inject yourself or a mouse's paw with wasabi, what happens is, of course, you feel pain. So you activate TRPA1 expressing nerve fibers. You activate the pain pathway by transmitting this information to second-order neurons in the spinal cord, and then ultimately this goes to your brain and tells you that you've experienced something painful. And you can detect this also by measuring expression of FOS in these second-order neurons in the spinal cord dorsal horn. But the other thing that happens, as I've talked about earlier, is that you get release of CGRP and substance P from the peripheral terminal, and this then leads to this process of neurogenic inflammation, namely swelling and reddening of the paw, that then enhances the sensitivity of this nerve fiber to temperature, touch, to more wasabi, etc. And this leads to a heightened pain response and initiates this process of inflammatory pain hypersensitivity. Now, if you inject the toxin into the paw of a mouse, you get a very similar acute pain behavior. You can see that if you inject Watox or the wasabi toxin into a paw of a mouse, you get what we call a nose-offensive behavioral response, or licking of the paw that's substantially greater than vehicle. And in animals that don't have the TRPA1 knockout animals, you see that they don't respond to the toxin. Similarly, you see an enhancement of CFOS expression in the spinal cord, showing that the toxin engages pain pathways, as you might expect. The surprise was that when you inject the toxin in, you get all these initial responses that you see with wasabi, but what you don't get is a release of peptides from the peripheral terminal. So there's no neurogenic inflammatory response because you're not releasing substance P or CGRP, and consequently you get acute pain, and you get a little bit of sensitization because you've probably driven this neuron so hard that you activate central systems that then become involved in pain hypersensitivity. But it's not, it takes longer for that to occur compared to wasabi itself. And we think that's because you're missing the neurogenic component of this process because you're not releasing peptides from the peripheral terminal. And we think the reason for that is because, and you can see that here, where injection of wasabi into the paw leads to a swelling of the paw if you measure thickness, but the toxin itself doesn't do that. And we think the reason for that is that you don't get CGRP release in the peripheral terminal. And in data that I won't show you, we believe the reason for that is because Watox, as I told you, does not allow as much calcium to come through the activated channel as wasabi itself. And this lack of calcium going through the channel probably is insufficient to drive the release of neuropeptide containing vesicles to release them from the peripheral terminal. So in summary, I hope what I've shown you is that natural products provide this great window into understanding the molecular mechanisms of somatosensation and pain. This wasabi receptor is really a unique receptor, plays a key role in detecting a very broad class of irritants that converge on this this common structural site that we call the cytoplasmic allosteric nexus. And wasabi actions are biophysically distinct, even though they work in sort of similar areas. And we're hoping that once we get a structure for the toxin on the channel and compare it to wasabi, that it will reveal distinct structural mechanisms that control ion selectivity. In particular, high permeability by calcium ions under some circumstances. I think this toxin's kind of cool. It sort of identifies a physiologic role for this novel toxin and for a new model for studying biology of membrane-penetrating peptides, which are still somewhat enigmatic. Mostly people focus on them because they're good ways, perhaps good molecules for drug delivery, but we really don't know much about their biology and how they work. So this is another model for studying that. And I think it also has helped us reveal what the importance is of high calcium permeability for these nociceptive trips. We've known for many years that many trip channels have a very high calcium permeability. We don't always know exactly what the physiological consequence of that is. I think what this toxin has helped us reveal is that one of the physiological consequences of this, at least for this nociceptive trip channel, is to be able to facilitate the release of peptide neurotransmitters from the peripheral terminal to support this all-important neurogenic inflammatory response. And then finally, so I just sort of mentioned this, and it also has provided this tool for us to be able to dissociate peripheral from central mechanisms that are involved in producing pain hypersensitivity. So it's kind of a novel drug for being able to dissect out the contribution of peripheral neurogenic inflammation from central mechanisms that drive pain hypersensitivity. And then finally, let me just once again mention the wonderful people who I've been privileged to work with on this. John King, recently graduated student who did all the work on the toxin and collaborated with Jinhua so beautifully to carry out the most recent structures of TRPA1. Candace Paulson now has her own lab at Yale. She and Jean-Paul started off the whole structural project on TRPA1. All the structural work's done with my great friend at UCSF, Yifan Cheng, and Josh Emmerich, a former student, contributed to some of the behavioral experiments I showed you at the end. Mark Kelly and Kati Mezratsky at UCSF helped us with NMR and mass spec analysis of peptides. And we've had this long-standing collaboration with Glenn King and Volker Herzig, University of Queensland, to obtain venoms from all types of nasty creatures, mostly insects. And all the work in my labs, or most of it, is supported by the National Institutes of Health. So thanks for your attention. Be happy to take some questions.
Thank you, David. That's a very fascinating and intriguing talk. Now I understand why I love wasabi.
Yeah.
I'll start. Probably it's because I can separate my periphery pain from my central pain. I might take the liberty to ask the first question. From your talk, I realize how actually all the different species on Earth, actually, we are well connected through the evolution. So, for instance, different toxins and different small molecule natural product may have different role on our neuron system through different channels. And this has been through a long time of evolution. So while I was listening to your talk, I was wondering, so it seems that only if the species have been, through the evolution have met each other, they might have some chances to evolve from a defense to anti-defense. If the species never met each other through I don't know if it would really be possible through the evolution. When two species, like two aliens, meet each other, they may not have actually much problem with each other because for instance, so would that be possible? For instance, if we meet something in the very deep sea, some new species, their natural product may not have any effect on our body, for instance.
I mean, yes. So an example in some ways of what you're alluding to, I think a nice example of that is to look at and their sensitivities, for example, to chili peppers, to capsaicin. And these are not species that have met each other, but that have different reasons for their interactions. So mammals, presumably, they like to eat chili peppers, and so the plant has evolved this, this molecule capsaicin that fits into a pocket that we think normally is there to appreciate endogenous activators of the channel, like bioactive lipids. But there are some animals on the planet in the same ecological niche that feed on the plant, but that actually, because their digestive system doesn't destroy the seeds, are actually vectors for dispersal of the seed. So while they destroy the plant, they're also beneficial to the plant because they're important for germination. Right. And those are birds. Right. So birds will eat chili peppers, and birds can eat chili peppers because they're not sensitive to capsaicin. And we showed years ago that there's a critical residue, which we can see very beautifully in the structure many years later, that packs up against the vanilloid in the binding pocket. Mm-hmm. It's this tyrosine 511, it's called in for example, the mouse structure, the rat structure. And that residue and residues around it, or that residue helps define a pocket that's structurally a little bit different in birds and makes birds insensitive to capsaicin. But the bird still uses the channel where it's still sensitive to heat and acid and other things that activate it. So there you see this sort of evolutionary, this evolutionary adaptation that's allowed birds to be insensitive to the capsaicin, so they're not averted from the plant. But the advantage for the plant is that the birds are good vectors for dispersal of the seeds. So there is a lot of very interesting examples of that in biology, so that's a little bit different answer to your question, but it shows you how, yes, there is rapid coevolution of species. And this is what I mean about if you compare different species for a given toxin, like the rat snake versus humans, right, this wasabi toxin, you can find you can quickly find differences, species differences, because there's a lot of adaptation and, niche adaptation. And, we as scientists can use those for structural studies very effectively.
Yep. I see there's a question from the audience asking, considering the TRIP channels are cation non-selective channel, do ions pass the channel in a hydrated or dehydrated form?
Yeah, that's a good question. There's not enough high-resolution structural data in all cases to answer that question, but where we do have some of the answers to this, it looks like that in many cases they're passing with a sort of partially dehydrated form. So they get partially dehydrated, the selectivity filter, and then they get rehydrated. Similar to potassium channels, but maybe not as strict a dehydration. And the selectivity filter, as I alluded to in most TRIP channels, is much more dynamic and sort of flexible. And it's not, you don't have this rigid, carbonyls that strip the water so efficiently from the ion. So in some cases, we think that the ions are going through is partially hydrated species.
Mm-hmm. Mm-hmm. Another question is, since TRPA1 V1 can also be activated by temperature, so what's the potential structural mechanism for the temperature sensitivity?
Right. Yeah. Well, temperature sensitivity is the big unanswered question in terms of structural biology. Mm-hmm. As you can imagine, it's a very tough one to answer. It's sort of analogous to the challenging questions that Ning is wrestling with in terms of trying to look at voltage sensitivity, but maybe even more so in a way. So we are trying some things to be able to trap channels in different states with temperature, but the whether and to what degree the gating mechanisms are and the conformational changes are similar to what you see with chemical agonists is just not yet known. And there are mutations in a lot of different parts of TRPV1 that affect temperature sensitivity. And so there's probably a lot of different places that are moving in response to temperature changes. We know that there are residues in the C-terminus that likely interact with the membrane and maybe impinge on the TRP helix that regulate temperature sensitivity. There are some in the outer pore region. I think we'll really need to see what structures at different temperatures to understand how all that information comes together. But that's a pretty technically tough challenge.
Could it be related to the lipid composition around the channel?
Yes, people have tried to explore that by putting the channel in different lipid environments. And there are no big changes in temperature threshold that seem to be mediated by changing the lipid composition, like putting in more or less cholesterol or other types of lipids. That doesn't seem to affect it so much. So that's probably not a big driver.
Yeah. Another one, since many TRP channels have a dilated pore size, do you think water can permeate through the channel as well?
Oh, yeah, that's a good question. You can see water in the pore of these channels. Whether it permeates in the same way that aquaporin permeates water, I don't know. And somebody else asked me that question, asked Yifan and I that question a few days ago. And that's, of course, a tough thing to measure. With aquaporins, you measure it by basically looking for swelling of cells. I don't know that anyone's looked at that. That's a very good question. You can see water molecules in the pore, so presumably they can permeate. Whether they're really permeating at any reasonable speed is nobody knows. And the same thing holds true for protons. There's been some suggestion that protons are permeant ions for some TRP channels, but it hasn't really been measured carefully.
Question asking is wasabi, garlic, cinnamon, all of them activate TRPA1, but why do they elicit different sensation?
Yeah, that's always a great question. I think in part it's because, it depends upon, to some degree, the volatility of these agents. Like when, wasabi is quite volatile. Of course, things like acrolein are very volatile. And I think in some part it depends not just how it's activating the channel, but which nerve fibers it's accessing when you come in contact with these things. So, for example, acrolein is something that's quite prevalent in things like vehicle exhaust or forest fires, and it's very volatile. And as a result, it will, or when you chop an onion, a lysine is the same way. It will absorb into the conjunctiva of your eyes and activate, TRPA1 on sensory nerve fibers in your cornea and in your eyes, as well as if you breathe it in. When you eat, some of it depends upon how we experience these things. Wasabi we usually take orally, so that's how we experience it. But I think the other way to experience it, which most of us don't do, but which is done sometime in pain tests, is rather than eating them or breathing them, is to just rub them on your arm. And if you do that, they actually have a sensation that's a lot more similar, because there it's really limited to a cutaneous pain and a cutaneous, and then a subsequent neurogenic inflammation. So, a lot of how we're experiencing these things is psychophysical, and how we take them in, and how we know that we're eating them or getting things. If you chop a chili pepper and you taste it, you get a very different sensation than if you forget you've chopped it and stick your finger in your eye, right? They're both painful, but in different ways. But if you do blinded psychophysical tests and rub these things on your hand, then you'll have a much more uniform experience.
Also, about a question about the Wa toxin, because it's a membrane penetrating toxin. So this question is about, is it too slow for preying purpose? For the prey. Oh, for prey. Yeah.
No, I think it's if you get stung by a scorpion, it's injecting this stuff into your skin, and the peptide. We haven't measured. Well, we have measured rates of access across a bilayer. We haven't measured rates of penetration across a cell, and I think to some degree it's going to be concentration dependent. But it is not slow. I mean, if you take the peptide or venom and put them on HEK293 cells in a dish, you see activation of the channel very quickly. Sub-second activation. So it can cross the membrane and get to its site. And, we don't know exactly how it gets to its site. So is it crossing the membrane and becoming soluble in the cytoplasm? Or is it just sort of flipping across the membrane? Remember, the part of the channel that's accessing it, this allosteric nexus, is very close to the inner leaflet of the membrane. And so it may not have to translocate all the way out of the bilayer. It may just have to flip across. But at least in patch clamp studies, you see it activate the channel very quickly. Maybe not quite as quickly as second time scale. So it's not something that takes minutes or, which is quite amazing, I think.
Thank you. If you don't have any more questions, I thank David again for your wonderful talk. And also, I see Woda is coming up.
Just to say good night.
It's pretty late. It's pretty late.
It's pretty late.
Yeah. Pretty late. Yeah.
And thank you, Ning and David, both for your wonderful talk today. And really, we all appreciate your time and your presentation. Beautiful. And hope we can see each other in person in the near future with all of us vaccinated. Yeah.
Absolutely.
Yeah, absolutely. Thank you. Thank you very much.
Looking forward to
you.
Thanks for pairing with me. That was a treat. So thanks very much.
Thank you. Okay. So this concludes today's session. And remember, everyone, we have our next session on May the 14th. And look forward to see each other again.
Okay. Thank you all. Good morning. Good night.
Good night.
Bye-bye.
Bye-bye.